Hi, good evening. Good evening, and welcome to the last Longwood seminar of 2012. We're delighted to have you here tonight. I'm Gina Vild, Associate Dean for Communications and External Relations and Chief Communications Officer at Harvard Medical School. Each week I mention that we encourage you to participate in 3 or more of our Longwood seminars, and you could earn a certificate of completion. This year, for the first time, we had 400 people eligible to receive certificates. So I want to thank you. Thank you. We're honored by your support of the Longwood seminars, and we're really grateful for your time. So I want to offer my applause to all of you. Thank you very much. Your certificates are available in the lobby. If you can pick it up after the program. So many of you have been with us, as we now know, week after week, but I do need to go through a few brief announcements. Each week, I tell those of you who are sitting in the front of the audience that you may show up on our YouTube channel in our video and to please change your seat. Well, I've never had anyone move, but I have to make sure that you know that you may be on our video if you are sitting at the front of the room. This year, for the first time, we're creating podcasts as well as videos from the series. These can be found on our HMS website, the HMS YouTube channels, and our iTunes U. Please share these links with your friends and your family who were unable to attend the seminar. As you also may know, our mini-med school is going green. So as a result, we have limited copies of the reading material, but you can find these online and they're easy to download. Similarly, our seminar survey will be sent to you via email. If you are not electronic and you don't have an email address, ask a volunteer for a print copy tonight. Teachers, as a reminder, you can earn professional development points by attending all 4 seminars and completing a form after each evaluation. These can be found at the table in the lobby. You can complete it here or mail it back to us. We hope you will ask our speakers questions, and to help you do this, you were given an index card. Members of our team will be wandering the aisles collecting the index card, so please write your questions. We also invite you to visit and to like our HMS Facebook page. It now houses the Longwood Seminars content. You'll find a tab for the new page called Mini Med School. Watch the site for more information on next year's events. We're also live tweeting this year. If you want to participate in the conversation live or afterwards, use hashtag HMSMiniMed. This is an extremely courteous audience, so I know I don't have to remind you to turn off your phone, but just in case, it's your reminder. And on a final note, behind the scenes of the Longwood seminars is one individual in particular who has been unwavering in her commitment to ensure the seminars are flawlessly executed. Katie DeBoff. Katie, come on up. Just, please. Yeah. Katie is a vital member of our communications and external relations team and has been organizing the seminars for the last couple of years. Katie introduced a high level of participant collaboration in these seminars. She makes sure that the audience is both an active member in choosing the programs and in evaluating each one. She's been indefatigable in her attention to detail. Katie, you've done a superb job once again this year, and on behalf of HMS and the members of this year's audience, we thank you for your creativity and energy.
Thank you. Oh, we have something here after you.
Thank you for all that you've done. Tonight's seminar is focused on how stem cells can be used to make us well. Harnessing the body's ability to repair itself, regenerative biology is advancing medicine each and every day. Our bodies contain stem cells that have the remarkable ability to transform into many different kinds of cells and act as tiny handymen to repair damaged tissue. Tonight, HMS faculty members will share their expertise and explain how this field of study may dramatically shape the future of medicine. Tonight's speakers are Dr. Chad Cohen, an assistant professor of stem cell and regenerative biology at Harvard, Dr. Fernando Campargo, an assistant professor of stem cell and regenerative biology at Harvard, and Brock Reeve. Brock is an executive director of the Harvard Stem Cell Institute. In partnership with the faculty directors, he has overall responsibility for the operation and strategy of the institute, whose mission is to use stem cells both as tools and therapies to understand and treat the root causes of leading degenerative diseases. He came to his role from the commercial sector with extensive experience in both management consulting and operations for technology-based companies with a focus on life science. Prior to Harvard, Mr. Reeve was COO and Managing Director of Life Science Insights, an IDC company, a consulting and market research firm specializing in information technology in the life sciences. We are delighted to have Mr. Reeve as our moderator tonight. Please welcome him.
Thank you, Gina, and thank you all for joining us here tonight. I noticed that we are under the auspices of the medical school and that you all have reading and homework assignments in your packets. But I thought I'd help you all out and just give you all that you need to know up front. So if you happen to fall asleep, it is late in the day, you're not paying attention, you don't want to ask a question later, all you need to know is this. Stem cells have only two characteristics that are important. They can self-renew and they can differentiate. So you need a cell that can do both those things, and that's all you need to know. Now, if you want to stay with us for the next hour and a half, we'll talk about a few. We'll talk about a few of the implications and what does that mean. A couple of the implications, and Gina alluded to those already, are not apparent at first glance. People early on thought about stem cells as therapies because they're aware of the cell ability to differentiate and to self-renew, which means to repair, to grow, to maintain the body. And we will talk about that. And one inherently sort of jumps to the notions of when cells aren't working, I can put new cells in. And we're all familiar with that. And we all know that cells do that because we've all donated blood. We wouldn't be able to donate blood if we didn't have stem cells. None of you would dare go get a haircut if you didn't have stem cells. And so we're familiar with some of the faster turnover systems of the body, blood, hair, et cetera, where stem cells are creating new cells all the time. We're less familiar with what we call sort of rarer populations. So we'll talk about some of those today. Because one of the efforts that has been going on over the last few years has been on understanding what I would call tissue-specific stem cells. In other words, stem cells that are related not, say, to the blood or the hair, as I mentioned, but cells are related, say, to the heart. Are there stem cells in the pancreas? In fact, we found out, no, there aren't. Are there stem cells in the liver? So how do different organs repair themselves in the course of adult life, but how do they grow in the first place? And we'll talk about what we've learned and what are some of the strategies there. We'll also talk about stem cells as targets because, and you say, what's a target? Well, rather than put a cell in a person, if we understand that there is a tissue-specific stem cell, there may be other ways to stimulate the body's ability to repair itself than just putting a cell in the body. You may be able to give the body a drug, a small molecule or biologic that can stimulate that rare progenitor cell population that may be resident, say, in the heart, for example. And we'll talk about some of those strategies. And then also stem cells as tools. And this is an underappreciated area because one of the things that I hope you'll take away from tonight is as we've learned how to create cells, how to grow embryonic stem cells and turn them into particular cells of interest, say a motor neuron, or how to take a skin cell from a person with a certain genetic disease, turn it into embryonic-like state, and then turn it into, say, a heart cell, for example. Or how do we take a mature neuron of one type and turn it into a mature neuron of another type? All that lets us manipulate cells, human cells, normal and disease, of particular tissues in the lab environment, in a petri dish. And what that lets us do for the first time is understand human disease in a way that we've never been able to before. And why is this interesting? For a couple reasons. One is we're all used to taking drugs. And a lot of the drugs, when they're brought to market, fail because they cause heart failure and liver failure, the two major causes, or they don't work on a major % of a population. And so one of the things that one could imagine doing, if we can grow heart cells at will for people with particular diseases, normal people, et cetera, imagine being able to test a drug on a human heart cell before you actually test it in a person. That's what a phase 1 clinical trial is today, right? It's testing on a live person. Wouldn't you rather do that in a petri dish, for example? Just last week, one of our colleagues, also a member of the Stem Cells, just published a paper on being able to do this, for example, for patients with cystic fibrosis, recreating human upper airway epithelium, multiple cell types, to understand outside of the human how the disease acts and how you can intervene in that disease process. So stem cells are the key to, we think, unlocking a lot of disease. They're key to understanding basic biology. They're key to understanding how we grow and how we develop and how we maintain ourselves and how we age. Because aging, for example, could be thought of as a loss of our stem cells over time. And so, for example, understanding how skeletal muscle repairs, because it repairs very differently in older people than it does in younger people. How can we understand the difference between those two processes, and can we therefore help older people repair muscle the way younger people can? So stem cells are opening up all these questions for us, and a lot of this is really due to a new understanding over the last decade of how malleable cells are. If you all remember the famous line from the Dustin Hoffman movie, The Graduate, where the guy tells him, remember one word, Benjamin, plastics. Plastics isn't the word today, but I would say plasticity is. So the one thing to remember is plasticity, that cells are flexible, and they're much more flexible and malleable and instructable with many fewer instructions than we thought even a few years ago. And I think you'll hear some of that tonight. So all you have to remember is self-renewing, differentiation, and plasticity, and you're all set. And then, but before we quiz you on whether you can remember 3 things or not, we'll have a couple of scientific talks from Chad Cowan and Fernando Camargo, and then we'll have a Q&A at the end. So I'd like to introduce Chad, who is, as mentioned, an assistant professor at Stem Cell and Regenerative Biology Department at Harvard, and also has a lab at MGH. And his research is focused on understanding how naturally occurring human genetic variation protects or predisposes people to cardiovascular and metabolic disease. Use that information to develop therapies that can, we hope, work. And so Chad's lab, both at Harvard and at MGH, is interested in using human pluripotent stem cells to create human-derived tissues, including specific DNA variants as genetic disease models. Chad was named a Stowers Medical Investigator in 2006, and he currently directs the Harvard Stem Cell Institute's IPS, that's Induced Pluripotent Stem Cell, core facility, and is head of our diabetes disease program. And tonight, Chad will be talking about embryonic stem cells as well as induced pluripotent stem cells. Chad.
Thank you, Brock, and thank you all for attending. I had not heard about the mini med school before this, but that's probably because I'm a PhD and never went to medical school. But I did have a passion for trying to understand disease, and I'm absolutely delighted to have the opportunity to tell you a little bit about the stem cells that I've been working with for, say, the past 5 or 6 years and actually 10 years here at Harvard. So the title, of course, as you can read, is Human Pluripotent Stem Cells, and then another concept that has become quite popular, reprogramming, and then, of course, their use potentially in regenerative medicine. So scientists love to repeat things so that you don't forget them, and I wanted to lay the ground rules again. When we talk about stem cells by definition have 2 very important properties. The first one, as Brock mentioned, is they have the ability to make themselves again, which we term self-renewal. And the second one, which is equally as important, is they have the ability to make a variety of different cell types or specialized cells. So they can make themselves or they can make other cell types. And it's through this way that we try to either harness their power to try to understand more about disease, or as my colleague Fernando Carmaga will tell you later, that's how you actually regenerate many of the tissues and organs in your body by having this one specialized cell make more of the cells that are either sort of sloughed off naturally or die or damaged throughout your life. So there are multiple sources of stem cells. You'll see as we go across the top from fertilization of the egg all the way to an adult organism, you can actually derive what are termed stem cells, or cells that have those two properties at different times in this sort of continuum. The earliest time that we've really identified stem cells are in these very early embryos. They're actually almost the size of a pinhead, much smaller than that even. And that's before they even implant into the uterus in most mammalian organisms or form something called a blastocyst, which we'll talk about later. And these are these embryonic stem cells that you hear about a lot. And the reason we're very fascinated with them is because we believe that they have the most potency or the most ability to make the most types of cells. As you extend sort of later into development, into fetal development, you can still derive cells, but we term these multipotent cells or adult stem cells. And then in the adult, of course, there are still stem cells often resident in the organs of choice, and these are either called tissue-specific stem cells, somatic stem cells, or adult stem cells. The big category that most of us use is adult stem cells. To give you an idea of what the two different types of stem cells do and why we categorize them as embryonic and adult is, in embryonic stem cells, as I mentioned, they have the most potential. They have the ability to turn into the most kinds of cells because they were taken from that very unspecialized early stage in development. They can, in essence, by definition, form all of the adult cell types, and we'll talk more about that. One of the other really interesting features of these is that they're considered immortal in culture. In other words, this ability to self-renew never seems to stop. I myself have never done the experiment to test whether or not they're immortal, but I can tell you that as long as we want to, we can keep them growing in the laboratory. So they continue to self-renew and replicate over time, making more and more of themselves. So that makes them rather plentiful. In fact, if we wanted to, there are companies out there that could fill this entire room with these types of embryonic stem cells. That's how many you could make. Adult stem cells, on the other hand, are very, very specialized. They're almost always organ-specific or tissue-specific. They also usually can only form a few cell types. In fact, they usually can only form the cell types that are found in their resident organ or tissue. So if you have a skin stem cell, it makes the cells that are in the skin. If you find a blood stem cell, also known as a hematopoietic stem cell, it makes all the cells that are in the blood system. They also have a limited lifespan. While they do self-renew quite a bit, they're only supposed to be with you for the term of your life. So sometimes these stem cells live a little bit longer than that, but they don't necessarily have this immortality or this ability to continually self-renew all the time. And then, of course, finally, they're relatively rare. They're usually the rarest cell in their particular tissue or organ. So they're sometimes hard to isolate or hard to find. But I think Fernando will be able to tell you some of the tools that we use to isolate them and identify them. And of course, we know that they're stem cells when they have the sort of two principles that I laid out at the beginning, their ability to make themselves again and then to make more cells. So I wanted to talk to you today more about embryonic stem cells and, of course, pluripotent stem cells. Embryonic stem cells were first derived in 1981 by Martin Evans. Martin Evans actually took this pre-implantation embryo, which has essentially two cell types. So if you see what's called this blastocyst, you'll see this outside cell type, and then you'll see this inside cell type. Scientists very cleverly called the inside cell type the inner cell mass. We're some of the most creative individuals on the planet. So the inner cell mass is that little ball of cells that almost looks like a grape cluster. Those cells are actually going to become all of the cells in the future adult organism. So in this case, in mouse embryonic stem cells, which are what Martin Evans actually identified, those cells are destined to become all of the cells in the adult mouse. The cells on the outside of that embryo actually become what is known as the placenta for that particular organism. So those aren't actually part of the adult organism. They contribute to sort of the development of the fetus. What Martin did was he actually isolated just those inner cell mass cells and put them in special conditions in culture in vitro, so in an incubator that was fed sort of a nutrient-rich media that allowed those to continue to grow and self-renew until eventually he had an established cell line, as we call it, so cells that were continuing to grow and grow and grow in culture, and these were embryonic stem cells. And the reason we were so fascinated with embryonic stem cells, and in fact, most of the research that occurred prior to us sort of understanding the potential of, say, a human version of this, was we could take these embryonic stem cells and put them right back into the exact same stage of development that we had taken them out of, and they would actually contribute back to the organism. So in this case, you could take stem cells, for instance, that were brown, and put them into a blastocyst embryo that had been taken from a white mouse. And later, they would contribute to all of the parts of that adult mouse, making what we would call a chimeric mouse, which would be both white and brown. So it would not just have, say, fur that was derived from the brown stem cells, it would actually also have all of its organs partially derived from those same stem cells. So this was the formal proof that stem cells of the embryonic nature could make all of the cell types of the adult body. And in fact, we used this technology to modify mice to make many of the sort of discoveries that you've heard about in terms of understanding which genes cause diseases, because we could actually manipulate stem cells to remove particular diseases that we knew, genes rather, that we knew were involved in diseases, and then make a mouse that actually lacked that gene and say, what happens to a mouse that, say, for instance, doesn't have the cystic fibrosis gene? Or in the discovery that actually led to the Nobel Prize that Martin and Evans and Mario Capecchi share, was the removal of the gene that is actually involved in a very debilitating disease called Leishmanians. They removed this one gene, made the mouse, and the mouse actually had a phenotype that looked very similar to humans that have Leishmanians. So it had the same sort of brain derangement, the same sort of very profound mental and intellectual disabilities. So this has been a very powerful tool. But what it led many scientists to think was, well, mice probably aren't the only organism that has this special type of embryonic stem cell. Perhaps it's possible for us to find this same type of embryonic stem cell in humans. And in fact, in 1998, James Thompson, working in Wisconsin, was the first person to actually isolate, in almost the exact same way Martin Evans did, a human embryonic stem cell. So you'll see sort of 4 pictures here. At the top is actually a human blastocyst embryo. Again, this is this pre-implantation embryo, much smaller than the pin of a head. In fact, if you plucked out a hair and cut it on end, you could fit about 10 of them in the sort of diameter that you would see there. And again, it's hard to see, but in that sort of hollowed out embryo is a set of inner cell mast cells. And you can see them isolated here on the right sort of lower panel. And those cells can then be plated again in this nutrient-rich culture media, and they give rise to these self-renewing cells that have the same potential as the inner cell mast cells that they're taken from. And you can see these little colonies over here on the left. Those are actually the growing inner cell mast cells that are now an established human embryonic stem cell line. And of course, why were we interested in human embryonic stem cell lines? Well, since we knew mouse embryonic stem cells had the ability to make all cell types, we postulated that the same might be true from human embryonic stem cells, that they could potentially make every single adult cell that makes up the human body, the supposed 260 to 280 very specialized cells that exist in all of our bodies. And in fact, through a number of experiments where we either guide the differentiation or allow the cells, these embryonic stem cells to just try to make any cell type, we found that they do make a lot of highly specialized cells. You can find that they make nerve cells like you'd find in your brain. They make blood cells. They even make these very specialized cells in your pancreas that are producing insulin that actually allow you to maintain your blood sugar levels and are affected often in diabetes. So we found that out, and you might ask, well, how is it that an embryonic stem cell makes one of these highly specialized cells? Well, one of the things that I think turns most people into believers when it comes to stem cells is seeing a picture like this. This is actually a culture of embryonic stem cells where we've teased them into thinking that they want to become a human cardiac myocyte, which is the cell that beats in your heart. It's your heart muscle. So if you look at that little green patch, you'll notice that it's spontaneously beating just like your heart. In fact, you might be able to even hold your hand up to your, take your pulse and find that that thing's beating at about the same rate. This happens either through a process that most of us try to harness in the lab, which is called directed differentiation or differentiation. Differentiation is the specialization of unspecialized cells. So those unspecialized embryonic cells in your embryo don't actually have a specialized purpose like a heart cell that has to beat and pump blood around the body. The specialization of those to something like a heart muscle cell, which you'll see just above that blood cell, is something that we term differentiation. And you'll ask yourself, well, how does this occur? In development, it occurs in a very slow sort of stepwise fashion, where you begin with an ICM or ES cell at the very top, and if you sort of follow this schematic with the arrows all the way down through the different colored balls which represent cells, you'll see that an ES cell can choose to become a specialized cell that will eventually give rise to all of the cells in your gut, all of the cells in your pancreas, lungs, et cetera, which we call an endodermal cell. Those cells can become further specialized to become the kind of cells that will eventually give rise to the pancreas. And then within the pancreas, there are subspecializations of cells, those cells that create digestive enzymes, and those cells that create some of the hormones that I spoke about, like insulin. And eventually you can become that cell type that only does, for instance, just insulin. And you might ask, how is it that these cells receive this information to become all of the different sort of in-between steps here? And what controls that is something you've already known about for a very long time: genes. So genes, or the DNA that are in the cells, and which genes are on and which genes are off, dictate exactly what your cellular identity is or what type of cell you are. So as you step down through this sort of differentiation cascade from an ES cell to a beta islet cell, you'll notice what I've schematically shown here with these little stoplights and go lights are genes that are either on and off at each particular stage. So in the endoderm cell, as it's called, you'll see in the top panel there's 2 particular genes on, but then there's a gene off, and then in the next one there's a gene on and the rest off. And it's this sort of code that changes as you go from one cell type to another that gives that cell its particular identity. You can think of it by analogy almost like its address. So initially you might get a zip code, then you get a very specific address, and then even within the house you know that you're in a particular room. And that's what the genes are doing. They're giving information. So by having a selective gene on or a selective gene off, that's what dictates function and specialization in those cells. So at the end of the day, what you can see is that if you compare cells to one another, you'll find that they have a very specific signature of genes that are on or off. So an example of that would be a nerve cell. You see the top row of genes is all on, whereas the next 2 rows are all off. And then there's some genes that all cells share. All cells have certain properties that are similar, so they're both on and off in all cells. But they have very specialized functions and thus very specialized genes that are on. For instance, nerve cells, as we know, have to conduct that sort of electrical signal to stimulate muscles, if it's, for instance, a motor neuron, and so they have to have a specialized set of genes on to make that specialized function. Blood cells, of course, are a little bit different, so they have a different set of genes that are unique to their own cellular identity that they turn on, and then the same is true for pancreatic beta cells. This same idea or concept is true for every single one of the specialized adult cells in your body, and it's this that we take advantage of as scientists, either to identify the type of cell that a stem cell has become, or identify a cell that we find in the body, or we take advantage of it to actually promote their differentiation into a specific cell type, like that beating heart muscle that I showed you before. So we can try to basically direct or teach them how to become a cell type that we're most interested in understanding. So again, just to recapitulate, the reason we're so interested in human embryonic stem cells, or what are now often termed human pluripotent stem cells, is their ability to become all of the adult cell types in the body, and what we think might be the ability to not just become that, but all of the tissues and organs that you find in the adult body if you could instruct them to that sort of sophisticated level. So I've told you about some of the endodermally derived organs, such as lung, liver, stomach, pancreas, and intestine, but there's also 2 whole other areas of differentiation: the ectoderm, which includes skin, brain, and nerve, and then mesoderm, which is blood, heart, kidney, muscle, and bone. But every cell type that you've ever heard of, you can make from a human embryonic stem cell, which is what got people really excited. So scientists, you could say, around 2000 got really excited about, okay, we've harnessed this potential for differentiation, and we've got human embryonic stem cells. How do we take this and bridge the gap to regenerative medicine? So on one side we had fundamental stem cell biology, and then on the other side we had what everyone sort of desired out of scientific endeavors: clinical care or therapies. How do you sort of fill that in? And we were a bit at a loss, because human embryonic stem cells had some limited potential and limited value, and they also had a lot of ethical concerns around them, because it required a human embryo in order to create them. So many scientists thought that maybe one of the ways that we might be able to avoid both the ethical concerns as well as some of the scientific concerns in using these for, say, transplantation therapies was the following, where we could try to do something which was turn back the clock on an adult cell to make it into something embryonic. So shown here is a diagram of what many researchers at the Harvard Stem Cell Institute around this time in 2003, 2004 were thinking about. Wouldn't it be fantastic if we could bring a patient who had gone to the hospital into the laboratory, remove, say, a skin cell or a blood cell from them, and then teach that skin or blood cell to become an embryonic stem cell, and then tell the embryonic stem cell, hey, why don't you make the liver cell that's damaged in this patient that brought them to the hospital for the first time and perhaps even transplant it back and thereby cure them. This was this whole idea of regenerative cell therapy. This was the concept that got many of us excited. And the exciting part about taking the cell from the patient and putting it back into the patient, as many of you know, it wouldn't be rejected. So when you need to get an organ transplant, you usually have to find someone who's matched to your organ type. Much like when you get a blood transfusion, you often need to find a matched blood type, or you have to get the type O negative universal donor. We thought we could avoid all of these complicated issues of trying to transplant cells from different people into different places, or these embryonic stem cells that might not be a match for a patient, by just taking a patient's cell, reprogramming it back into a patient-specific pluripotent cell, and then using that for these cell-based therapies. At the time, of course, you might say, well, why did you even think this was possible? Why did you think you could sort of turn back this developmental clock on a cell that was already adult and make it into something that was less than adult? We'd been taught through a couple of very fascinating experiments that it was in fact possible. The one that many of you have probably heard about was Dolly the sheep. So Ian Wilmot in Scotland had actually cloned a sheep. He took an adult cell from the breast tissue of this sheep, put it into an enucleated egg, and then activated that egg, and it used the DNA from the adult cell to basically go all the way through development to make an exact genetic identical copy of Dolly the sheep. So Dolly had a little baby that was basically Dolly's genetic copy. And that's shown on the right. And the process by which that's done is called nuclear transfer, where you take a somatic cell or an adult cell, remove the nucleus, and put it into an egg that's had its genetic material removed, and it can go back through development. If you were to let it go through development but pause it at the embryonic stage and then use that embryo to make stem cells, you would in fact be able to make a patient-specific stem cell. The disadvantage I think you can already start to see is that in order to complete this process in humans, you have to use human eggs, right? So we knew that the process only worked when you took these nuclei and put them into a human egg, and that's what sort of rewound the clock on them. Again, that had a lot of ethical concerns around it because there's only one source of human eggs that we know of. In addition, there were a lot of technical concerns that went along with this. And to this day, it's never been successfully accomplished. So that led us to sort of think outside the box. What else could we do? Well, one of the ideas that was floating around out there was, why not just take a human embryonic stem cell that's already been made and see if you fuse it to an adult cell, what would happen? So basically, fuse the two cells together so that you're making a single cell out of them, and in that wrestling match, what happens to the resulting cell? What we found, much to our surprise, was what you generate is a cell that looks almost identical to a human embryonic stem cell. This fused cell that has DNA from both cell types now thinks it's a human embryonic stem cell. So that taught us one very important lesson. It taught us that everything you need to accomplish this sort of rewinding of the clock exists in an embryonic stem cell. So that led a lot of scientists to do what we always do best, which is a reductive approach. Start to pull apart all of the things that are specific to embryonic stem cells that we might be able to add back to adult cells to give them back these special properties of pluripotency, the ability to make all the cell types, and of course self-renewal. And it wasn't long before there was a huge breakthrough that we were all quite stunned by. So in 2006, Shinya Yamanaka, working in Japan, was able to introduce just 4 very specific genes that are found in embryonic stem cells into actually a fibroblast cell, the same kind of cell that you find in your skin. And he made it into something that was almost identical to a mouse embryonic stem cell. The whole world just sort of stopped. I remember when he gave this presentation scientifically at a meeting that was in Canada, all of us walked out sort of shaking our heads like, this is impossible. It can't be that simple. Like 4 genes that come from a stem cell into a fibroblast cell and you've got a stem cell again. It turns out through many sort of iterations and more research, we found that they're in essence functionally indistinguishable. You can use those same cells, put them back into say a blastocyst stage embryo, and it will make one of these chimeric mice again. They'll differentiate into all the adult cell types. So we were able to do what is in essence rewinding of this clock or reprogramming of cellular identity by doing just what I told you before, changing the genes that were expressed in the cell. These key genes were able to sort of set up a hierarchical cascade of gene expression that made the cell now look identical to an embryonic stem cell. And these so-called induced pluripotent stem cells have been sort of the watershed by which we've been trying to use cells to understand more about therapeutics. So now what we think we can do, and in fact can do, is go to almost any person in this room or any patient that comes to the hospital, and then with defined factors reprogram an adult cell, and often we use a blood cell and make it back into a patient-specific pluripotent stem cell. And then now the idea is still out there that we could maybe differentiate those cells into a cell that might be damaged as a result of disease and then transplant it back into the patient. This is the idea that drives a lot of Parkinson's research, some diabetes research, et cetera. But what you'll notice is at the bottom there I've written another thing which is called in vitro models. So the other idea is that you might be able to use these to learn something about the disease that brought the person to the hospital in the first place. And you might develop not necessarily new cells to put into them or new organs to put into them, but something that we're all much more familiar with, which are in fact probably new drugs to treat that particular disability. So the Harvard Stem Cell Institute has been very busy in this area, and what we've been trying to do is make what we would consider disease models. So we've taken patients that have a variety of diseases, and we've actually made these induced pluripotent stem cells from them so that the community at large could in fact start doing research to learn more about Parkinson's, Huntington's, muscular dystrophy, Down syndrome, you name the disease. In fact, the induced pluripotent stem cell core that I currently direct has made over 400 different disease-induced pluripotent stem cell lines that they've distributed to researchers across the world who are doing a lot of interesting research, and I'll sort of outline what they're thinking about. So two of the sort of big publications that describe these induced pluripotent stem cells are shown here, one by our friend George Daly here at the medical school and another by another colleague, Kevin Egan, which was actually looking at Lou Gehrig's disease. So the idea is fairly simple. In my laboratory, we care about diabetes, the consequence of obesity. So the idea might be that we could take an obese patient who's got diabetes and derive through this reprogramming with defined factors a patient-specific pluripotent stem cell, and then of course differentiate that cell into fat. And if you were my mother, you'd say, why would you want to make more fat, Chad? Well, the reason that we use stem cells to make cells that we want to study in a dish is that usually it's relatively difficult to get them. Like, you can't ever get a neuron from somebody because they want to use them, they don't want to give them to you. Most people volunteer really quickly to give you some fat for your research in the lab. I know, because we work on it. The problem is, even after you've given me fat, I can't keep it alive very long in a dish. It doesn't have that special property of self-renewal. Fat actually is the end stage in that organ, so you only get those fat cells to do an experiment on one time. And almost everybody's like, I'll come back, I'll give you more fat. It turns out you can't give me enough fat to do the type of research that we want to do, so you've got to be able to have a renewable source of these human cells to try to understand the disease. And let me give you an example of what I might want to do in terms of, say, obesity or diabetes. So you can imagine now, I'd have a disease-specific induced pluripotent stem cell from somebody who might just have, say, obesity, or somebody who might actually have, say, insulin-resistant diabetes or diabetes. And then you have cells from people that don't have it. Maybe it's a sibling or a parent that doesn't have the disease but is very closely related. You could say, turn these, as I mentioned, into fat cells, and then compare the two fat cells. Say, what's different about the fat cell from the person who's obese as compared to, say, a normal person, or the person with diabetes as compared to the normal person? And what we hope to find in these cells that are affected in the disease is some dysfunction, something slightly different about that cell that you might be able to repair. In other words, some deficiency in the cell that you could maybe repair and then turn that cell back into the healthy cell. And the way we look at it, of course, is by looking at all the genes that are expressed again. We look at the functions that fat cells are supposed to do, which is like storing fat, releasing fat, listening to insulin, taking up glucose. So this is a very powerful tool to really get down to the, exactly what's working in a disease and how does it go wrong. And I think, as I mentioned, and as even Brock mentioned, the therapies of today that are going to come from stem cells aren't us turning a stem cell into a whole new organ and transplanting it into someone. It's going to be the same sort of therapies you're used to. It's going to be drugs. Because since I can make an almost unlimited source of these stem cells, once I've identified something wrong with the fat cell that's differentiated from that, you can basically run what is called a small sort of drug screen. Look for a drug that repairs that deficiency in the fat cell, and that drug might actually eventually become a drug that you could use to treat the particular disability. Let's say it's diabetes, or maybe even in my mom's fantasy, obesity. So thank you very much for your attention. I hope I explained to you a little bit about why we're so excited about pluripotent stem cells.
Thank you, Chad. And you weren't supposed to be recruiting donors for the lab, but it's all for a good cause, believe me. Now welcome Fernando Camargo. Dr. Camargo is also an assistant professor at Harvard and has a lab in Children's Hospital right around the corner here as part of the stem cell program there. Fernando's ultimate goal is to understand the signals, some of which Chad has alluded to, that regulate adult stem cell maturation and tissue regeneration for their eventual manipulation and application in the clinic. His lab has been a pioneer in the novel biochemical cascade that controls stem cell numbers and activity. And one of the corollaries of that is that it regulates, he's looking at how to understand organ size and growth. Because one of the interesting questions is, as things grow, how do they know when to stop? Fernando has been awarded NIH New Innovator Award, named a Basil O'Connor Scholar from the March of Dimes, and is also a Pew Scholar in the Biomedical Sciences. So this evening he'll be focusing his talk on adult stem cells, which aren't only resident in adults, and transdifferentiation as well. Fernando.
All right. Well, it is an absolute pleasure to be here tonight. Out of all the talks that I give, I really get the most excited about giving talks to audiences such as the general community, such as yourselves, because at the end of the day, all the work that we do and all the impact that we want to, the clinical impact that we want to get to, is something that would be useful for all of us, including most of you. And also, I remember I gave a talk at the Museum of Science several years ago, about 7 years ago, and I had a question from, I think a teenager. He was 14 or 15, not a scientist. And I was just talking about how blood stem cells grow, right, and they replicate themselves, and you have this massive capacity for regeneration. But then he asked me, so if they grow so much, then how can they stop? And that's not really very well characterized. So from that question, my lab has really developed an almost entire area of research. So I really appreciate the opportunity to discuss some of these issues with you guys, not only because of the general interest that I have in your input, but also because I think scientifically there might be something fantastic coming out of the discussion that we're going to have in a few minutes. Okay? So I have to thank Chad for the fantastic introduction that he's given to the stem cell field. I will be telling you a bit about the other stem cells that he referred to, the stem cells that are derived from adult tissues, right? And I just want to make a few points before we start discussing some of the main issues that I want you to take home. And so at some point in our lives, we've all been and we've all resembled that cell on the left, right? We all have gone through that stage, and our progeny will all go through that stage. That's a single cell, a single very unspecialized cell, a mother stem cell, the mother of all, the totipotent stem cell, okay, very unspecialized. And as is evident through series of cellular divisions and complex process of cellular differentiation and specialization, you end up with an adult that has hundreds of different cell types, each one with a unique and different function, okay? So this issue of cellular progression, of assuming more specialized characteristics is going to be a common theme throughout my talk, okay? So I want you to keep that in mind, and this also highlights the potential differences between embryonic-derived cells, which are more akin to the ones in the left, and cells derived from adult tissue, right, which would be more equivalent to the ones to the right. And again, this is just maybe a bit a different way of showing you this slide before, right? During human development, there is a hierarchy of cells, and a hierarchy that is associated with not only maturation, development, aging, if you want to call it, growth, but it's also associated with the potential capacity of some of these cells to give rise to more cells, to different types of cells, right? So whereas very early, you have these totipotent embryos that can give rise to a full organism, and then also embryonic stem cells that Chad told you about that in theory have the capacity to give rise to most of the tissues in a human. As development proceeds, then you have a specification, right, determination into particular sets of lineages that give rise to only a certain number of tissues, right? So as development, as growth proceeds, you get this restriction in developmental capacity, right? So they're not as potent, they're not as plastic. We're going to talk about this later. And also, as growth and development proceeds, you get this increase, you gain differentiation characteristics, Okay, so these are very common points that we should take home. And again, in the adult here, then you have several different tissues, several different organs, and again, each one of those has a unique function, okay? Again, stem cells. I mean, Brock already told you all that you need to know about the general idea behind stem cells. They're unspecialized, and this applies to that very early embryonic stem cell. This also applies to all the stem cells that we have currently working really hard in our bodies, okay? And we'll see a few of those. Another sort of requirement for these cells to be named stem cells is the fact that they can produce more stem cells, right? This is absolutely key, right? It's like the mothership, right? It produces more progeny, more smaller ships, but also it can produce more of itself. It can retain itself. It's long-lived, okay, long-term. They can also produce specialized cells, right? So they can make more of their own kind, but then at the same time give daughters that have those unique functional capacities, okay? Okay, so let's talk about adult stem cells. Brock already mentioned some of those, but here I just want to highlight really the remarkable degree of capacity for these cells to sustain growth, to regenerate. So every day, you get 500 billion cells, blood cells, produced in your body, right? And these cells are produced in your bone marrow, right? In the cavities in your bones all throughout your body. And these cells are not only red cells, these are platelets that help with blood clotting. These cells are immune cells that fight pathogens and bacteria. So there's this constant renewal. In the intestine, right, this is your gut, a small intestine, large intestine, 100 billion cells are also produced every single day, right? Imagine this massive capacity to produce cells. In the skin, again, another highly renewing organ. So if somebody asks you, can we regenerate? And the question is, at least in a homeostatic level, just to maintain our bodies as such, we can, right? And there's a very interesting area of investigation looking at whether these processes of normal regeneration are, for instance, reduced with aging and some sort of pathological conditions. But the bottom line is that we have stem cells. We have these very remarkable cells that allow long-term regeneration and sustainment of tissue renewal, okay? So there are other stem cells in other tissues. I'm not going to have time to discuss some of them. And there are some tissues that can regenerate well, not as well. Muscle, for instance, right? Muscle can regenerate, not as much and not as fast as some of these tissues. It's been even proposed and demonstrated that for the brain, there are stem cells in the brain. The question there is still what the function of those stem cells are, right? Because you would imagine that if you have stem cells residing in the brain, if you have an injury in your central nervous system, you should in theory be able to regenerate. It is a bit unclear why that does not happen if you have stem cells. And now if we look at a bit closer scale here at some of these stem cell compartments, you find remarkably the exact same hierarchical organization that I show you in that developing human embryo. Okay, so this is looking at the cells that are within your bone marrow. Again, these are the cells that produce all the blood, platelets, and immune cells that we have. And this is basically all the cells that are in our blood and our marrow, right? All of these cells, and these cells are the specialized cells. These are your red cells, your white cells, your pathogen-fighting cells. They all come from one single cell, a very rare cell in the bone marrow. So if you take a bone marrow, and you sort single cells, and if you look in the microscope, how many of those are actually stem cells? How many of those can actually transplant? How many of those can actually give rise to all of these cells? It's only 1 in about 100,000, okay, or less. So these cells are very, very rare. But again, as they divide, they lose their capacity to give rise to more cells, right? You have these stem cells, but you have these daughter stem cells that give rise to stem cells that are committed, can only make 1 or 2 groups of cells, but not all of the cells, right? So this sort of progressive restrictive capacity is also present in the adult, right? This hierarchical organization is also present in the adult. I'll give you another example, the gut, right? We talked about also its high capacity for regeneration. This is a scheme of basically what you have in the lumen of your small intestine, right? You have these finger-like projections that go inside your tube These are called villi, and these are the projections that actually take up the nutrients that come from when you have a meal, and are being taken up by this. But as you can see here, there's also these invaginations here, right, of these villi. And these invaginations into the stroma here are called crypts, right? And these crypts are the anatomical location where those stem cells reside, okay? So stem cells are not just hanging around everywhere in the villi. There is a unique, particular location of these stem cells here at the very, very bottom, okay? And if you zoom that in, you see that your stem cells also within that anatomical niche, anatomical place, they are in contact with a neighboring cell. And you don't need to know the name of this cell, but this neighboring cell is crucial to make the stem cell happy. If you don't have this neighboring cell here, you have no stem cells and you lose capacity to regenerate, okay? So basically your stem cells need to be in a particular anatomical location, likely because they talk with other cells. They need to be in the right environment. They need to be in the right mix. They need to have the right juice to be stem cells, right? And that is really what we're trying to understand with our work. And we'll talk a little bit more about that. Okay, so again, let's talk about the difference between ES cells, embryonic stem cells, and adult stem cells. Chad already talked to you about these. What I want to discuss today is two issues related to that are very interesting and very intriguing and perhaps somewhat controversial. So, I'm very open to have a lively discussion in the next 10 minutes. So, one of the attributes that have been attributed to having given to adult stem cells is that they cannot really be expanded indefinitely, at least as much as embryonic stem cells, right? That's why it makes embryonic stem cells very, very attractive. I'm going to tell you about some recent work that people have done in which they've demonstrated that it's in fact possible to expand to almost unlimited quantity some stem cells, which it really marks a breakthrough in our ability to work with adult stem cells. And also the other issue I want to start discussing right now is whether, again, another dogma, which you will probably understand now from the figures that I show you of that progressive restriction of developmental capacity, is that these adult stem cells are only restricted in their ability to give rise to cells of the tissue in which they came from, right? Liver stem cells, you'd only give rise to liver. Heart stem cells, you'd only give rise to heart. Okay, so I want to have a small discussion about that, which I think is quite relevant, especially for some of the some clinical applications that are currently ongoing. Before getting to that directly, I just want to highlight very briefly that stem cell therapies are being used right now, right? So it's not just a promise. These therapies have been used since the 1960s. And the biggest demonstration of this is the work that has been done with hematopoietic stem cells, right? The cells that reside in your bone marrow. So these cells reside in your bone marrow, and you can transplant them. You can isolate bone marrow from one donor, and then you can simply inject those cells intravenously into a recipient. You guys may have known people, friends, family members that have undergone bone marrow transplantation, peripheral blood stem cell transplantation, right? So this is a widely used protocol, and it saved thousands and thousands of lives. So you can take bone marrow stem cells, you can transplant them, and this is highly effective. There are clinical protocols to do this. This is very, very highly effective. So this is really the gold standard, at least right now, for adult stem cell therapies. And we can discuss some of the issues that have actually made this particular model, the bone marrow model, to be so efficient and so successful in the clinic. And this can be used for many conditions, right? And at this point, all of the conditions that are used, we can talk more about this, cancer, some genetic conditions, are conditions whose problem is mainly at the level of the bone marrow, at the level of the blood system, okay? Again, blood stem cells help, and in some cases cure diseases of the blood, okay? And this was actually recognized by a Nobel Prize, Donald Thomas at the Fred Hutchinson Center in Seattle several years ago for the first application of stem cell transplantation in patients. Okay, so this issue of whether stem cells have this restricted capacity, right? Whether a cell from the liver would only give rise to a cell from the liver. This was highly debated and discussed back in the early 2000s. And the findings were that many scientists all across the US and the world were finding that if you were to transplant, say, a neural stem cell into the bone marrow, or a bone marrow stem cell into the brain, you would see that that stem cell, initially thought to be restricted to the site of origin, would then give rise to cells of a different tissue, right, of the tissue in which you had placed them on. Right? So this opened up this entire novel possibility that the potential of these stem cells normally is restricted by their physical location, right? So that if you were to take a bone marrow stem cell and move it around and take it to the muscle, then the bone marrow stem cell will act as a muscle stem cell, which was quite remarkable. And again, went against the dogma of what you have probably been taught in your biology classes, definitely what I have been taught in my biology classes. And this had several implications, right? And I'm just showing you some of these, but you can think that you can envision, for instance, the use of transplantation of bone marrow that I just described to you, which is a highly used clinical procedure for the treatment of muscular dystrophies, for instance, right? You would never thought of that before. Injection of bone marrow stem cells for the treatment of cardiac regeneration. Bone marrow or other cell injections for neural diseases, right? You had all these potential avenues for therapy. However, over the past several years, work done by many scientists, including some of our own work, has demonstrated that these observations really do not hold very rigorous scientific evaluation. Right? It is true that maybe a bone marrow stem cell will give rise to a liver cell or a heart cell, but it's not something perhaps very reproducible and physiologically relevant. And more importantly, there's no very strong demonstration that, for instance, transplantation of bone marrow cells into the hearts of infarctic patients would actually help them. Right? So the majority of the scientific community would argue that there isn't really any strong proof that injecting, say, stem cells from the marrow would help other tissue, okay? Despite of this, though, and this is what I want you to discuss when you're at home and having dinner, is that not only in the U.S. but all around the world, people have taken some of these early discoveries and they're really trying to profit from them, perhaps from from a science that is not based on very rigorous results. So if you look for stem cell therapies, for instance, in Google, you can find that basically there are centers in Taiwan, in Thailand, in the Dominican Republic that promise to put stem cells into, say, brain if you have Parkinson's, and in many other cases of diseases. Right? Again, there's no scientific proof that that's the case, but there are thousands of dollars and many, many patients still doing this. Even in Texas recently, this was a feature in Nature magazine just a few weeks ago. There's a company there that is actually injecting fat-derived stem cells, right, into patients for all kinds of diseases, including multiple sclerosis, Parkinson's. Again, there's no scientific proof that this is working. So if somebody offers you to do that, really think about that twice and talk to somebody that could probably give you a better insight. So this is really a bit of a controversial aspect of the potential of the stem cells. Okay, so we can discuss more about that later. So I just want to then highlight those some advances in how now people are thinking about adult stem cells and how we think now it is actually possible to actually expand these cells, right? And the key to do this has been the understanding of that basic biology, understanding how those stem cells form basically in the embryo, right? In that hierarchy that I show you, where those stem cells come from, right? What other cells is this stem cell close to, right? What is the juice? What are the signals that keep the stem cell happy in that microenvironment? So people have done this over the past years, and there's been very exciting results, and I just want to share two of these with you. And this work actually comes from the work of a colleague here, actually at Children's Hospital, also at the HSCI, and they actually study blood stem cells, interestingly, in the zebrafish. Okay, so fish are also very good for biomedical research. So what they do is they study thousands of zebrafish embryos, not even adult fish, just zebrafish embryos. And they put the zebrafish embryos in chemical screens. And they can find some chemicals that actually induce the formation of blood stem cells in a 3-day-old embryo. And you would think that doesn't really have much relevance, right? But then they took some of the chemicals that did this and put into adult stem cells, and they actually did the same. They expanded the number of those stem cells, and they increased the ability of those stem cells to engraft into recipients, right, after bone marrow transplants. So remarkably, since this has worked so well, they've actually been able to now start clinical trials with very successful results with this molecule, right, indicating that you can actually manipulate the ability of these stem cells to function properly and in some cases be expanded. Another very exciting development comes from a lab in the Netherlands. And what they did is they've developed a model. Sorry, you don't see much there. They developed a method in which you can actually expand intestinal stem cells. This is something that nobody has been able to do before until just 3 years ago or 4 years ago when this study was published. So how they did it is they identified all the signals that are required for that stem cell in the intestine to be happy, right? Years of work, 10, 15 years of work. And then they took every single one of those components, they put them in the Petri dish, and they put intestinal cells. Just a simple biopsy of a human intestine even can do this. They can put them in the Petri dish, and these intestines actually grow indefinitely. You can actually put the dish of the intestine in the freezer, come back a month later, take it out, and it will still keep growing, almost forever. So this is really a breakthrough. And remarkably, what happens in the Petri dish is that you not only get stem cells growing, you actually get stem cells and also differentiated specialized cells growing as well. For instance, this is just a picture of those cultures, and these are mini organs, basically, right? These are mini guts that grow in the dish. And you can see here that the stem cells are in the green here, and they're producing all those other differentiated cells here in the gut. Right, so you have these mini guts just going crazy, growing and growing. At some point you need to break them up because they get too big. So it's quite remarkable. So we, for instance, in the lab have been able to adapt this culture, and we can grow mini liver organoids in the lab. So you can expand those liver organoids forever. So you can think about the very exciting possibilities to do with those. And again, this is a scheme of those mini guts. Again, it has 2 domains. It has those script domains that I told you, those invaginations that you normally see in your gut. And they basically recapitulate that organization. And they also has this lumen, right? This basically hole that resembles the tube that you have within your gut. So this is really quite exciting. And applying these same sort of culture techniques to other stem cell populations that are harder to culture, such as pancreas, for instance, if there's a stem cell, or neural stem cells, I think that's really going to be quite, I think, groundbreaking. The other exciting thing that this allows you to think about now is to actually grow organs in vitro, right? If you think about an organ, it's a complicated structure, right? Getting cells, say the cells that make up the hepatocytes, getting a bunch of them, you can do, right? And we can do with those stem cells. The question is, how do you integrate them into a 3D structure that would actually work and that potentially you could implant into a patient? One of the questions is solved, right? We can get now an unlimited number of cells, either through this expansion of intestines or livers, or through embryonic stem cells. And the next question, this is something we're doing in the lab now, is what else do you need to get that architecture, right? So bioengineers now are working on ways of generating that frame, right? Think about a house, right? The stem cells and the cells produced by the stem cells are the bricks or the blocks that are put around the house, but the actual structure, the actual core, right, of this structure, it's composed by other components, right? Together with bioengineers, now you can generate these sort of frames, and then you can think about using your stem cells to put them into those frames, and then to actually make an organ. So this is now a possibility. So with that, I'm just going to end one more slide, and that is that the study of adult stem cells now, the exciting aspect of this, it's revealed that you can learn a lot about cancer from studying adult stem cells, right? All that juice, all those molecules that keep the stem cells, for instance, in the gut, very, very happy, also happen to be extremely present, omnipresent in all cases of colon cancer, right? So the molecules that make these stem cells to be long-lived, to be able to give rise to billions of cells a day, are also are also those molecules that allow your tumor cells to just keep growing and growing and growing, and to recur after chemotherapy. Okay, so this has been very exciting, and so that's another reason why studying adult stem cells could be, therapeutically beneficial. With that, I think I've went above my time, and with that, I will end. And thank you so much for your time.
So thank you, Fernando and Chad, and thank you all for your questions. And we have a bunch, and I'm glad that you finished on the cancer slide, because there were a couple of cancer questions for Fernando. But before we come back to cancer, let me, because I was called to task here. Someone actually was paying attention to what I said, and we'll give Chad a question on diabetes, because I mentioned in passing that there wasn't a stem cell in the pancreas. And so what does that mean for the role of stem cells in diabetes, particularly type 1 diabetes? So Chad, can you talk about what we do as a result, and how can we therefore intervene in diabetes for both type 1 and type 2?
Yeah, so thanks, Brock. As we've looked into more and more organs after the discovery of sort of the first tissue stem cell, which was the blood stem cell, we've discovered that some organs don't actually maybe have a stem cell resident in them to allow them to replenish those cells that might be lost. There's another even simpler way in which they actually replenish cells that die or lost, and that's just by dividing. The cells that are already there just divide to make new cells. So in the case of diabetes, when you have type 1 diabetes, there's an immune attack that kills the cells that produce insulin, so these insulin-producing beta cells. When we looked at what makes an insulin-producing beta cell in an adult, it's actually insulin-producing beta cells. They just divide to make more of themselves very slowly over time. So that meant that we couldn't isolate a very special stem cell from that organ to make new, like transplant it into the organ to make new insulin-producing cells. But what I told you today was we knew that embryonic stem cells or pluripotent stem cells had the ability to make any cell type. So we thought that there might be the opportunity to make insulin-producing cells from this. And there had already been clinical trials done where people had actually harvested insulin-producing beta cells, as they're known, from cadavers, so people who had died in car accidents, for instance, or motorcycle accidents, which is the major source of organ donation. And they would collect these and actually put them into patients who had no insulin-producing cells in a standard transplant, and it would actually cure them of their diabetes temporarily until the immune attack came back and started eating the cells. So I think there's the idea out there that this stem cell replacement therapy could happen, that we could take a pluripotent cell, make the cell that's been lost or damaged as a disease, and in this case an insulin-producing beta cell, and transplant it back into the patient, just as done before with real adult cells.
Thanks. Thanks for digging me out of that hole, Chad. Fernando, let's come back to the cancer question. We had several questions about cancer. First of all, if stem cells can keep growing, do they potentially turn cancerous? Second of all, how do we think of the cancer stem cell, and if we look at tumors, can any cell in a tumor become cancerous? What's the relationship between stem cells and cancers, and is that different maybe in solid tissue tumors than it is, say, in blood tumors, leukemias?
Right, so the question deals with cancer stem cells. So there's two issues, right? The first question is whether cancer arises in the stem cells of a tissue, right? And I think most of these issues still haven't been clearly worked out, but you would think that it's very, very logical, right? That a cell that is long-lived, a cell that will be in your body for life, could acquire a mutation, right? And then still, the progeny of that stem cell still be hanging around for your whole life. So that increases the probability that a second or third mutation occur in that same cell, or the lineage of that cell, so then you develop a tumor, right? So, and for cases of blood malignancies, acute leukemias, for instance, it has been demonstrated that some of the mutations occur in the stem cell compartment or very early stem cell progenitor compartments.
It's not been directly demonstrated, at least in the mouse. It has been demonstrated in solid tissues, for instance, in the intestine. But it does not necessarily mean that all the mutations happen in the stem cells, right? Another way to look at this would be that all the tumors, maybe if they're not derived from stem cells, as they develop into tumors, then they hijack the molecular identity of stem cells, right? Because stem cells have this machinery that tells them, okay, keep them going, we need to grow. Tumors may hijack those, and that's also been shown. But I think the bottom line here is that these same genes, these same signals that fuel stem cell growth, that fuel regeneration, in many, many cases are highly involved in cancer, right? And the particular case is colon cancer. You need this set of signals called WNTs that if you don't have them, your intestinal stem cells cannot grow. And you basically, in mice, for instance, if you disrupt this, your mice die because the intestine completely disappears. And colorectal cancer cells, in patients, 90% of them have mutations in these WNT molecules. Okay, so the understanding of this set of molecules that regulate stem cell biology, stem cell identity, is going to be absolutely crucial, and that has been demonstrated all over and over again. So I think the issue is going to be how do you target tumors distinctly from stem cells, right? Some people argue that tumors maybe need more of these signals, right? So there is a selectivity there. But I mean, these are very interesting, I think, and just novel questions that people are trying to answer.
So, Chad, related to that, we have questions sort of related to reprogramming and iPS cells. And so let me combine several of them into one big question. One is related to the whole reprogramming process. Can iPS cells become cancerous, particularly once implanted? Second of all, in the reprogramming process, what is the role of, you mentioned that they aren't the same as embryonic stem cells, what's the role of epigenetics? And one of the methods that you mentioned was cell fusion. And doesn't that mean that you'd have two sets of DNA, and isn't that not what you want?
Yeah.
So related to the whole...
I'll do those in reverse order.
Combine them all in one.
The cell fusion was really one of these experiments that was purely of a scientific basis, where we wanted to know who won if you made a stem cell and an adult cell sort of battle about their cellular identity by fusing them together. As the questioner noted, they do have both sets of DNA, so they're not useful for therapy. You couldn't put them back into someone or use them to create a new heart muscle cell, et cetera. So that really just taught us something fundamental about cellular identity and the fact that stem cells had something in them that would allow you to sort of almost transfer stem cell qualities to a different adult cell. In terms of the first question, which was cancer, about iPS cells and cancer, it turns out that if you take embryonic cells and put them in an adult environment, many different types of embryonic cells, they have a continual growth program that's incompatible with where you might put them, so they often do form tumors. In fact, embryonic stem cells and induced pluripotent stem cells, if not differentiated, if put back into an organism, make a tumor, usually sort of a benign tumor known as a teratoma. These are these funny tumors that have teeth and hair, bones, etc., but also sometimes invasive, aggressive tumors. So that's one of the risks and one of the things that scientists think about when trying to use these for transplantation therapies, because when we want to differentiate them into a specialized cell type, you want to make sure there's none of those undifferentiated stem cells left over, because they have the possibility of causing a tumor or a cancer. But there's no evidence that a fully differentiated mature cell has that same capacity unless it's accumulated some of the mutations Fernando talked about. And what was my number two that I forgot?
Epigenetics.
So hard quiz. Oh, epigenetics.
And
So
the
what's
difference
different between an IPS cell and an ES cell? There's a lot of things that you could imagine. So I talked about some of the genes that are expressed, et cetera. There's other layers that sort of govern what genes are on and what genes are off. And this is a thing that we call epigenetics, which are the things that are actually interacting with the genome or the DNA as it's compacted into sort of the chromosomes. Those things that turn on and off genes and sort of are sitting around genes are often a little bit different between induced pluripotent stem cells and embryonic stem cells, and we don't fully understand it. What is interesting is that at least in the animal models where we can study this rigorously, the function of the cells seems to be almost identical. Although it depends on whether or not you've fully reprogrammed. In other words, turned the clock all the way back and made something that really, really resembles an embryonic stem cell. Sometimes you've done this less successfully, so it's a little bit confused and stuck in this sort of partial state, and then it doesn't have all of the right stuff to make either a differentiated cell or always continue to self-renew. So it's still very early days, but I think that the idea that epigenetics was one of the things that might be different is in fact correct, and there's even some evidence that the cell type that you take it from might actually, the stem cell might remember where it came from. So in other words, if you started with a blood cell to make an iPS cell, it might still remember that it was once a blood cell, so it might more likely make blood cells. So this idea of epigenetic memory is also prevalent, and there's a lot of research going on into it. But I can tell you that at least in the mouse, where we can study it very rigorously, induced pluripotent stem cells function identically to embryonic stem cells that we make from embryos.
Okay, so now let's venture into an area that neither one of you are experts in. And
Chad will take that one.
okay, so Fernando, thank you for volunteering. A couple questions relating to Alzheimer's. We've talked about diabetes, we've talked about obesity, we've talked about, what about a complex disease like Alzheimer's? How are stem cells useful either as tools for helping us understand the disease or as potential therapies? Because one of the things one hears about in the press, and should that be in the list of diseases one thinks about as subject for or likely candidates for stem cell therapies?
Yeah, again, I think Chad would definitely answer this question better from his expertise. But I think that the real advantage of using iPS cells is the ability of studying diseases for which there is no mouse model. Diseases that are polygenic, meaning that it's not only one mutation, but several mutations, complex diseases, right? And so Alzheimer's, I think some forms fall in that category. So in theory, you could make cells, you could make neurons that from those patients, right? And the idea is that I think that's the question, if you can actually see some sort of problem with the neurons derived from these patients compared to the neurons derived from normal patients, then you can study the disease. In theory, you should be able to. I think the challenge is to make this specific type of cells that show this problem in patients, and I think it's possible, right? I think the other issue is with modeling diseases as well, and probably Chad has a lot more about this than I have, is that we're thinking about these diseases to be at least maybe perhaps too simplistically, right? Perhaps we're thinking that Alzheimer's only affects this unique type of neuron, right? And if you want to model that disease, if it's just one neuron, one type of neuron, then yeah, you can do it. But the way I think about it is that perhaps in some situations you're going to have a disease that affects more than one cell type. Right, so if you want to model that correctly in the dish, you would need to make 1, 2, 3, 4 different cell types and put them in the same concentrations or percentages, so you can actually mimic something. So I think the idea is there that can be done, but I think there are some challenges as well. So I don't know if you want to add something to that.
Related to that, maybe Chad, you can comment on, for example, Kevin's work, and taking up on Fernando's comment, maybe Kevin Egan's work on ALS with the glial cells.
Yeah, I think that what the questioner highlighted is one of the real challenges that biology's faced in trying to understand disease, is it's not always as simple as one gene has a mutation and equals one disease. It's often a combination of maybe many small mutations that don't have a large effect, and then environmental influences, so things that we've done throughout our life. Like, it's no surprise often if you've drank too much throughout the course of your life that you've gotten liver disease, or if you've smoked your entire life that you might get lung disease. That's an environmental insult that leads to a particular disease. But it's interesting, you'll sometimes see in those smokers, some people will smoke for their entire life and never get cancer. So they don't have these susceptibility factors that somebody else who unfortunately smoked smoked for only 5 years and got a very aggressive cancer. So we're starting to understand that there's genetic predispositions for particular diseases. And in the case of ALS or Alzheimer's, we're starting to explore that as well. There are genes that give you certain susceptibilities for these. So you can have small changes or variants that you're carrying around that give you an increased risk for a particular disease or protect you against that particular disease. In the best cases, what we're doing as scientists is taking still those really rare forms, which are inherited where we know a gene is mutated and it seems to cause this disease, and it looks a lot like, say, everyday Lou Gehrig's disease, ALS, or Alzheimer's. Those we can very easily make a stem cell from that patient, and then that stem cell into, say, maybe the neuron. And in ALS, or Lou Gehrig's disease, we know one particular cell type almost always dies. It's this motor neuron, the neuron that actually transmits signals to your muscle. So we can take patients who've got these inherited forms of Lou Gehrig's disease, make a stem cell from them, and then turn that stem cell into a motor neuron and say what's happening. And I think why we're really excited as scientists about this opportunity is we've never even been able to study human motor neurons or human cortical neurons, let alone like a more complex organ. So we really think that just by looking at the cell type that actually is affected in the disease, we're going to learn something about it. Even if it's just plain vanilla Alzheimer's that doesn't have a genetic influence, there might be some defect in the cell that leads us towards what's actually happening in the disease. To be quite honest, there's only been a few breakthroughs, and they've come from all of these rare genetic diseases. We now understand some of the ways in which those lead to Lou Gehrig's disease, some other rare familial diseases like familial dysautonomia, other inherited diseases that affect neurological development, even some inherited forms of autism like Rhett's disease.
And if I actually combine part of Fernando's last comment and part of Chad's last comment, one of our colleagues at the Stem Cell Institute was looking at ALS, at Lou Gehrig's disease, and normally you think it's a failure of the motor neurons, which is true. But then when you say what causes the failure of the motor neurons, and he showed that at least in some cases, it's the surrounding cells to the motor neurons, these glial cells, that cause normal motor neurons to fail and to go bad. So if you thought, well, all I have to do is replace the motor neurons, you'd be wrong, you wouldn't have solved the problem. And so the notion that Fernando was talking to about the multicellular environment that the cells live in, we had to be careful about not being too reductionist in our in vitro approaches because these cells are talking to one another. And in this ALS example is, you could be a good person living in a bad neighborhood and the bad neighborhood was going to have an effect on you. Unless you can and it's hard for the one person to turn the neighborhood around in that case. Totally different question. To go out on a limb here. If stem cells can create new cells and therefore, we're talking about affect the aging process and affect healthy aging, what does that mean for what we can think about in terms of life expectancy, and what does that mean in terms of society, how we think about stem cells affecting longevity, and then you can sort of expand to whether it's healthcare costs, whether it's, people want to live forever, or where you want to go with that. So what are the social implications of some of this?
Well, I mean, I'll take one stab at it. Usually when I think about using stem cells as therapies or adding benefit for degenerative diseases, I don't think any of us really think we might, well, maybe some of us do, but most of us, and in particular myself, wouldn't want to live forever. I just want to feel like I'm 18 until I'm 80. So if you can actually... So if you can actually regenerate those tissues so that
already,
yeah,
Chad.
it's true. Let's just put pause now. But it would be fantastic if, when I got out of bed, I wasn't creaking around the house all day. So I think that's the real concept here is you could regenerate or renew it, but you're not necessarily going to stop or forestall death, for instance. And if you think about it that way so that you have healthy aging or healthy organisms up to a certain finite point, that's a more rational way. I think no one necessarily wants to die, but all life on the planet has a beginning and an end. And so if you think of healthy aging or making sure that you are as healthy as possible until that end, that's probably the best way to think about it and not have a huge impact on society. I'm not sure if you've got a different thought, Fernando.
Yes, actually I do want to live until I'm 250. So, I mean, first of all, I don't think we're there yet in that we could either, activate endogenous programs of repair in our aging tissues that would alleviate some of the aging problems and phenotypes that we see. We're not there in terms of making organs out of scratch. I mean, I do. So it's. I mean, some issues that that are related to aging are just beginning to be understood. I mean, there's probably a very likely, and it has been demonstrated for some tissues, a stem cell component, right? And interestingly, it really seems that perhaps it might not be the actual stem cells per se that have aged, right? But it's that microenvironment, right? It's, for instance, something in your blood that is making your cells age. This colleague in our department, Amy Wager, she actually showed very nicely in these crazy experiments that she actually put together an old mouse and a young mouse. And don't ask me how, but she put them together, and this mouse actually shares circulation, right? So the blood of the old mouse circulated into the blood system of the young mouse and vice versa. So what you actually saw was that exposure of young blood, basically, right? That's really literally the way to call it. Into the old tissues, it basically rejuvenated them, which is remarkable, right? But that speaks that it might not be the actual stem cells themselves, but it's something outside, right? Again, this environment is juice. So I mean, I think at this point it's premature to talk about that. I think we're not quite there yet, but. There's also some implications. I mean, with aging, cancer also arises, right? The more time we live, the higher probability of our cells to acquire mutations and develop cancer. So I mean, it's a complicated question, full of financial, ethical dilemmas. I don't think we're there yet. I reserve my opinion for the next 20 years.
And many more evenings worth of discussion. A couple of sort of social policy related questions. Comments about any thoughts you guys have on federal regulations as well as, for example, state funding efforts like the state of California or New York. And how does that then also, related to that, how does that impact sort of how the U.S. competes in this field, say, compared to other countries, and what are some other countries doing in terms of stem cell research?
I think that probably refers mostly to embryonic stem cells. So maybe many of the audience recalls the presidential order that was signed by former President Bush, which restricted the use of federal funds for embryonic stem cell research. He drew sort of a bright line in the sand that said any embryonic stem cells made before this date are fine, any embryonic stem cell made after that date not fine, even though they're the same cell, but it was an interesting policy move. Unfortunately, when he chose to draw the line, he thought he was making a fairly informed decision, like, okay, we've got enough stem cells over here in this bucket that have been made for us to do some research on. And it looked like from the scientific reports there were something like 32 available. It turned out after the end of that there were about 8 that you could get your hands on if you were a reasonable sort of scientist and you went to the ends of the earth. Since then, the politics have evolved. So under Obama, any embryonic stem cell line can actually apply for inclusion in the National Institutes of Health, which is the main funding body for biology registry. And then if it's registered because it's been created through the appropriate ethical permissions and the right channels, you can then use federal funding for that particular stem cell. It's the regulations are the regulations. In reaction to those restrictions, we saw many states start funding stem cell research on their own so that we could try to sort of achieve this promise. And interestingly, I just had a discussion earlier with a student who was doing a research paper on the history of some of this in my office today. And it turns out that the impact was bigger than I think a lot of people felt. In Harvard, we sort of swim in a rarefied air. There was enough private funding for many of us to continue to do the research we did. For instance, myself, and a mixture of private funds from Harvard and private donation from people who had given to the Harvard Stem Cell Institute, continued to do the science that I thought was the best science that would answer a question. If you went someplace else, like where I got my undergraduate degree, like the University of Kansas, they never even started doing embryonic stem cell research. And the reason was the guidelines were so confusing during the Bush doctrine that they couldn't understand if they had NIH funding to do one type of science but started to do this other type of science, whether it would contaminate it and whether they would have to shut down the science that was legitimately funded versus the other. There wasn't a good way of sort of separating it. And how complicated it is is when I get a federal grant from the government, it actually doesn't just pay for me to buy reagents to do research or for the salaries of the scientists, it pays for the lights, it pays for the maintenance people. There's these costs built in for those grants to sort of keep the building going. So you can imagine if I did 2% of the research we did was with embryonic stem cells and they weren't approved by this federal, what 2% of that would be the maintenance people and the rest. So some of these universities said, we can't figure this out. We don't even have the lawyers that have the time or the energy, and nor do we have the money. We're not even going to get started. So the ripple effect was most state organizations and state schools didn't even get involved in stem cell research. And we thought for a long time this would put us at a massive disadvantage at stem cell research and there would be a flock to Europe. I will say that Europe definitely, which has not had as large a stem cell research community previously, caught up in almost no time. So it's a great group. You saw some of the work from Hans Cleavers, whether it's adult or embryonic. And it's maybe no surprise that the biggest sort of breakthrough in the last 5 to 10 years in stem cells might have come from Japan, where it's a much more liberal environment for doing embryonic stem cell research. The good news is we're back with induced pluripotent stem cells, which give us none of the ethical quandaries and have none of the constraints in terms of funding. We're really sort of charging ahead in terms of disease research. But the regulations did change the tenor, and I think it drove some of maybe the best and brightest young scientists who thought, why would I want to go into this murky field where it's not clear that I'm going to get funding and it's not clear that you're going to be able to do anything, to make choices that were quite different. So.
Any closing remarks, Fernando? We're about
I just say that's the reason I stuck to adult stem cells.
May they all stick with us too. I know we're at the end of our official time, but do stay and chat if you'd like to. And I apologize we didn't get to as many of your questions, but please come down and talk to people. And we look forward to continuing the conversation not only today, but in the future as well. Thank you very much.